Hello, good evening to the students who have joined. I hope my voice is audible. Please let me know if my voice is audible and you can see the video. Thank you for confirming. Can you please let me know if the presentation slide is visible to you? Is the PowerPoint presentation visible? So I hope the presentation is visible and you can hear me properly. So we will wait till it's 6 p.m. for we will wait for 5 more minutes and then we will start today's session. Okay. Meanwhile, if you have any doubt or any query regarding our last class and anything, you can let me know. Any doubts or any query regarding our previous session or previous class, which was the week 9 live session, you can let me know now. Okay. We have still 5 more minutes left till we start the session. Okay, so it's 6 p.m. I think I should start the session. Meanwhile, when more students will join, they will eventually join in because the call is open. So I hope my voice is audible and everything is all okay with the presentation. If not, please let me know. So, very good evening to the students who have joined. My name is Mousumi Deb and I'm a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for this NPTEL SWAYAM course of Medicinal Chemistry, and today it is our week 10 live session. So, before starting the session, like every day, I would like to clarify a few things that this kind of problem solving sessions are scheduled on every Monday from 6 to 8 p.m. Okay, these sessions are live on Google Meet platform, and this is the link for the particular Google Meet for joining the class. Okay, you can use it, you can copy this Google Meet link and you can use it for joining every Monday class. Okay, if any network disruption happen from my side, do not leave the session immediately. I will try to rejoin as soon as possible. And the call is open. So if you have any issue with your network, you can rejoin back at any time without asking permissions. Okay. Since I'm recording the session, I have muted your microphones and cameras, but the chat box is always open. So if you have any doubt, any query, anything, you can let me know through the chat box. Okay. Every now and then I am going to check the chat box. So if you have any problem, any doubt or query, you can let me know through the chat box. If any doubt persists after class, you can drop an email to me. I will share my email ID with you at the end of the session. Also, you can write your queries in the discussion forum of this SWAYAM portal, and this presentation and class recording, it will be shared to you after the end of the session. Usually I try to share this particular slide and the recording that day itself, and if it is not possible, I will surely give it to the next class. Okay, so yeah, good evening to all of you. So where to find this problem solving session material, I have already told you many times, but let me share you one more time. So you have to go to the SWAYAM portal. There you will find, on this is the announcement page of the medicinal chemistry course. Now, on the left panel, you will find all the weekly updates of the course material, whatever is uploaded each and every week, along with the quizzes and everything. Okay, there at the end, you will find one thing which is known as the problem, which is the problem solving session, January 2026. If you click on that, you will find that problem solving session recording, and clicking on this page, it will take you to a page which looks like this. It has all the YouTube link for the each and every class, okay, recording, and also it has the Google Drive link. This Google Drive contains all the presentation whatever we have completed. So till now we have completed 9 classes, so there are 9 PPTs here. Okay, so you can download the PPTs from this page and from this Google Drive, and then you can read the things whatever we have completed. So, okay, if you have any doubt or any query during the class, you can let me know through the chat box. After the class, you can let me know your doubts or queries via email. I will share my email ID at the end, and also you can post your queries in the here. There is a Q&A, which is the discussion forum. You can write your queries there also. Okay, whatever is okay with you, you can let me know. So, as I have already mentioned, today it is our week 10 live session. So, like every live session, how we do it, I usually show you around 20 MCQ question. Each and every question have 4 different answer. After showing you a question, I will wait for some time, and during that time, you have to let me know in the chat box of that A, B, C, or D, whichever which of the following will be the correct answer for that particular question. Okay. After a minute or two, I will check the chat box and I will check your answer. Then I will show you what is the correct answer and we will discuss it in detail. Okay, so how I have designed the class, like the MCQ questions will help you to understand, to help you to solve your assignments, which is the weekly assignments, and the detailed description of each and every question will help you to understand that particular topic in detail. Also, it will help you during your final examination. Okay, so this is what we have to complete. So I hope everything is fine with the audio and the presentation. So let me just start the session. Okay, so today it is our week 10 live session. Let me show you what is the course content. So this is the course content of our week 10. It is a first we will discuss a little bit about the combinatorial chemistry and parallel synthesis, then computer and medicinal chemistry, then we will discuss about the antibacterial agents. Okay, so in our last class, as if I remember correctly, we have discussed a lot about the prodrugs. Okay, the prodrug, why prodrugs are important, what is the designing of the prodrug product, what are the important things that you have to take care of when you are designing a product for a particular therapeutic. Okay, so these things we have discussed. So from now on, in our remaining classes, we are going to discuss about various therapeutic agents. Like in today's class, majorly we will be discussing about various antibacterial agents. In our upcoming class, I think we will be going to discuss about the particular antiviral agents and also anti-cancer agents. And in our last class, we have something which is known as your anti-ulcer agents. Okay, so various therapeutic agents. So from now on we will be focusing on that part of medicinal chemistry. Till now we have learned what are the basics of the medicinal chemistry. Now the application part. Okay, so that we are going to discuss. And one more thing, it is our 12-week course. So today it is a week 10 live session. Apart from today's class, we will be having two regular classes, the week 11 and week 12, and one summary class that I will take on I think I will take it on 14th of April, which is on Tuesday from 4 p.m. Okay, the exact schedule will be shared to you via email about the timing and the class link and the date also. So at the end, I will take a summary session to summarize the complete course and to discuss a few topics which I think needs to be discussed one more time. That we will do it. Okay. So let me start the session with the first question of today's session. So the first question of today's session, it says that Medifil resin is, now, then there is a dash. Okay, so you have to identify which of the following is the Medifil resin. Your options are number A, chloroformylated polystyrene, number B, chloroacylated polystyrene, number C, silicon dioxide, the polymer of silicon dioxide, and number D, polymethyl methacrylate. Medifil resin is which of the following? Chloroformylated polystyrene, chloroacylated polystyrene, the polymer of silicon dioxide, or polymethyl methacrylate. Which of the following will be the correct answer for problem number 1? I will wait for a minute or then I will check the chat box. Okay. Meanwhile, let me know which of the following you think is the correct answer for problem number 1. I have mentioned it a couple of times that during the session it is important for you to answer these questions, okay? Because there is no negative marking, nothing is there, so you don't have to worry about anything. But try to answer the question, otherwise I won't understand that you are following the class or not, okay? Which of the following is the correct answer for problem number 1? What is Medifil resin? Number A, chloroformylated polystyrene. Number B, chloroacylated polystyrene. Number C, polysilicon dioxide. And number D, polymethyl methacrylate. Which of the following is the correct answer? Let me now check the chat box. Okay, I have received two answers and both are saying that A is the correct answer for problem number 1. Now let me check if A is the correct answer or not. So yeah, the correct answer is number A, chloroformylated polystyrene is the Medifil resin. This is the structure of Medifil resin. Okay, so this is the polystyrene and this is the chloroformylated polystyrene basically. So this is the how the particular Medifil resin is denoted. Okay, so the first successful example. So this is a part of the solid phase synthesis. Now, what is the solid phase synthesis? I will discuss it in detail in our upcoming slides again. Okay, so basically in the solid phase synthesis, the first successful example of solid phase synthesis was the Medifil resin peptide synthesis. Okay, so the resin involved consisted of a polystyrene beads where the styrene is partially cross-linked with 1% of divinyl benzene. The beads are derivatized with a chloromethyl group, the anchor or linker, to which the amino acids can couple now via an ester group. This ester group is stable to the reaction condition and used in a peptide synthesis, but can be cleaved off at the end of the synthesis using vigorous acid condition like with the hydrofluoric acid. So this is our resin bed, this is our Medifil resin. Now in case of the solid phase peptides, so how the peptide synthesis happens in the solution phase, I think we have discussed it a couple of times in our earlier classes. I mean in our first or second classes we have discussed it couple of times. Okay. So this is basically an amino acid with in this particular amino acid, this N side is protected with a Bock protection, okay, and the carboxylic side it is open. So now this carboxylic acid it will react in this position and it will form a ester bond, okay, and then after that this particular N protected amine is deprotected with the TFA or trifluoroacidic acid. Then what happened? Then another amino acid which is the second amino acid it comes which also have this N protected and this carboxylic end is open, and now the peptide bond is formed like this, okay, and after repeating this particular reaction for couple of time we have this our polypeptide chain, okay, but it is attached with the resin bed or to the solid phase, okay, that is why it is known as a solid phase synthesis, where this particular peptide chain is synthesized while it is bound to a solid phase, okay. At the end, how to cleave this particular thing, you have to do a vigorous acidic workup with a concentrate with a strong acid like hydrofluoric acid, then you will have this particular polypeptide chain and you will get back this thing, okay. So this is how the peptide synthesis on a solid support happens, okay. Here Bock is the tertiary butyl oxycarbonyl group and TF is the trifluoroacidic acid I have already mentioned a couple of times, okay. So they remember this is the Medifil resin which is the chloroformylated polystyrene, and it is very important because it is vigorously used in the solid phase peptide synthesis. And how this solid phase peptide synthesis happen, if you are asked, then you have to give this particular diagram, okay. So this it is very simple, you have to just practice it a couple of times, okay, then you can easily draw it if it is asked in the examination part, okay. So I hope it is clear. If you have any problem, please let me know, okay, in the chat box. Please let me know if I have to repeat any question. Then I will go to the next problem. Problem number 2 say which of the following statements regarding solid state synthesis is false. Your options are number A cross-linked insoluble polymeric support is needed. Number B, an anchor or linker should be attached with the support. Number C, the anchor should contain reactive functional group. Number D, the protective group of the functional groups should involve in the synthetic routes. Which of the following statements regarding solid state synthesis is false? Again, I will repeat the statements. Number A cross-linked insoluble polymeric support is needed. Number B, an anchor or linker should be attached with the support. Number C, the anchor should contain reactive functional group. Number D, the protective group of the functional group should involve in the synthetic routes. Which of the following statement is false regarding the solid phase synthesis? I'm going to wait for a minute, then I will check the chat box, read the options very carefully. And like I already mentioned it couple of times that whenever there is a true false kind of question, read the statements very carefully because sometimes these answers or these questions can be really confusing. Okay, read all the options and let me know. Number A cross-linked insoluble polymeric support is needed. Number B, an anchor or linker should be attached with the support. Number C, the anchor should contain reactive functional group. Number D, the protective group of the functional group should involve in the synthetic routes. Which of the following statement regarding solid state synthesis is false? Let me check the chat box. Let me see if there is any answer. Okay, I have received two answers, and okay, I have actually received three answers, in which one is saying number B and other two is saying that number D is correct answer. Okay, let me check which of the following will be the correct answer for our problem number two. So the correct answer will be yes, very good. Number D. The statement which is false is number D, which says that the protective group or functional group should be involved in the synthetic groups. So, solid phase techniques can be used to carry out reactions where the starting materials is linked to a solid support such as the resin bed. Several reactions can be carried out in sequence and on the attached molecule. The final structure is then detached from the solid support. Okay, so there are several advantages. So, whenever we are discussing about the solid phase synthesis, so there are several advantages to this since so what are the advantages? Since the starting material, intermediate and the final product are bound to the solid support, excess reagents or unbound byproducts from each reaction can be easily removed by washing the resin. Okay, the large excess of the reagents can be used to drive the reaction to completion greater than 99% because of the ease with the excess reagent can be removed. Intermediates in the reaction sequence are bound to the bead and do not need to be purified. The polymeric support can be regenerated and reused if the appropriate cleavage conditions and suitable anchor or linker groups are chosen. Automation is possible. If a combinatorial synthesis is being carried out, a range of different starting materials can bound to the separate beads. The beads can be mixed together such that all the starting materials are treated with another reagent in a single experiment. The starting materials and the products are still physically distinct as they are bound to the separate beads. In most cases, mixing all the starting materials together in the solution chemistry is a recipe of disaster with polymerizations and side reaction in a terrace. The individual beads can be separated at the end of the experiment to give individual products. Okay, so these are the advantages of the solid phase technique. So now the essential requirements for the solid phase synthesis are, so what are the things that are required while we are doing the solid phase synthesis? Number one is the cross-linked insoluble polymeric support, which is inert to the synthetic condition. For example, a resin bed that we have discussed, like our Merrifield resin is one of the such example. An anchor or linker covalently linked to the resin. The anchor has a reactive functional group that can be used to attach the substrate. A bond linking the substrate to the linker, which will be stable to the reaction condition used in the synthesis. A means of cleaving the product or the intermediates from the linker. Protecting groups for the functional groups not involved in the synthetic groups. Okay, so that the number four, the what is it is said that the protecting groups for the functional groups not involved in the synthetic group. Now let's go back to our options. So number one, a cross-linked insoluble polymeric support is needed. This is true. Number B, an anchor or linker should be attached with the support. This is also true. Number C, the anchor should have certain function reactive group, which is also true. Number D, which says that the protective group of the functional group should involve in the synthetic groups. This is false because here we can clearly see that it is written that the protecting groups of the functional groups are not involved in the synthetic group. So these are the requirements for the solid phase synthesis and these are the advantages of the solid phase synthesis. Okay. So I hope it is clear. So do I need to repeat it or is it okay? Let me know if there is any doubt or any query. Okay, I hope it is okay. Otherwise, let me know. Okay. If there is any issue, I will repeat that particular question whenever you get the you do not understand. So now let me go to the next problem. So problem number 3 says which of the following statements regarding molecular mechanics is false? Your options are number A. In molecular mechanics, the molecules is treated as a series of spheres connected by the spring. Number B. Torsional energies are associated with atoms separated from each other by 3 bonds. Number C. Molecular mechanics is used to calculate the electronic properties of the molecule. Number D. Molecular mechanics follow the classical laws of physics. Which of the following statements regarding molecular mechanics is false? Again, I will repeat number A. In molecular mechanics, the molecule is treated as a series of spheres connected by spring. Number B. Torsional energies are associated with atoms separated from each other by 3 bonds. Number C. Molecular mechanics is used to calculate the electronic properties of the molecule. Number D. Molecular mechanics follow the classical laws of physics. Which of the following statements regarding molecular mechanics is false? Let me know A, B, C or D, which of the statement is false regarding molecular mechanics. Again, I will wait for a minute, then I will check the chat box. Read all the options carefully and then let me know A, B, C or D, which of the following will be the correct answer. Which of the following statements regarding molecular mechanics is false? Number A, in molecular mechanics, the molecule is treated as a series of spheres connected by springs. Number B, torsional energies are associated with atoms separated from each other by three bonds. Number C, molecular mechanics is used to calculate the electronic properties of the molecule. Number D, molecular mechanics follow the classical law of physics. Let me check the chat box. Let's see if there is any answer for our problem number 3. Okay. I have received two answers and both are saying that number C is the correct answer. Means number C is the statement which is false regarding the molecular mechanics, right? Okay, let me check. So the correct answer, the statement which is false will be number C. Molecular mechanics is used to calculate electronic properties of the molecule. This statement is false. Now, molecular mechanics is a computational method that uses laws of classical Newtonian physics to model the structures and the properties of the molecules. Okay, unlike quantum mechanics, which treats the molecules as the systems of nuclei and electron, molecular mechanics ignores the electrons and treats the atom and the individual spherical particles connected by springs representing the chemical bonds. So, our first statement, which says that molecular mechanics, the molecule is treated as a series of spheres connected by springs. This is true, and also the number D, which says molecular mechanics follow the classical laws of physics, which is also true, right? So core principle, what are the core principle? The method relies on the force field and empirical set of energy functions and parameters that define the potential energy of a molecule system as a function of atomic coordinates. Balls and spring model, atoms are treated as the masses are balls and bonds are springs. Total energy or potential energy calculated as the sum of several energy terms. One is a bonded interaction, energy from the bond stretching, angle bending and torsional dihedral rotation. Non-bonded interactions, energy from van der Waals forces or steric effects, and electrostatic interaction, Coulombic force between the partial charges. Parameterization. The specific constant used to in the equation is the equilibrium bond length spring constant are derived from the experimental data like X-ray crystallography and IR spectroscopy or high level quantum calculations. So common force field, different force fields are optimized for the specific types of systems and not generally compatible with one another. For example, we have this amber AMBER, which is widely used for proteins and nucleic acid. We have this charm, which is popular for modeling macromolecules and biological system. Bromos, often used in molecular dynamics for biomolecule. ALPS, which is optimized for liquid simulation and organic molecules. MM2, MM3, MM4 developed by Norman Ellinger primarily for small organic molecule. What is the key application? The energy minimization, finding the most stable or lowest energy conformation of the molecule, molecular dynamics, simulating the movement of atoms over time and solving the Newton's equation of motion. Drug discovery used in protein ligand docking to predict how the drugs bind with the target, and hybrid quantum mechanics and molecular mechanics, combining quantum mechanics with the molecular mechanics for the large protein environment. So these are the key applications. So this molecular mechanics actually is not used for the calculation of the electronic properties of the molecules. Okay, and so this statement number C is false. So this is all about the molecular mechanics. We will be discussing regarding the molecular mechanics in detail in our upcoming slides. Okay, along with the quantum mechanics. Now let me go to the next slide. Okay, so what are the major limitations? Let us discuss about the major limitations also. So, number one is the no bond breaking or forming. Standard molecular mechanics cannot model chemical reaction because bond connectivity is usually fixed. Electronic properties, because electrons are not explicit. Molecular mechanics cannot predict like UV spectra, magnetism, or electronic excited state. That is the electronic properties of the molecule. Empirical dependence, the accuracy of entirely dependent on the quality of parameters for the specific molecule being studied. Okay, so these limitations are very important. Okay, so what you have to remember basically, so is the core principles. Okay, and also the major limitations of the molecular mechanics, like it cannot predict the bond breaking or bond forming, it cannot predict the electronic properties, and also it has some empirical dependence. Okay, because of the quality of the parameters of the specific molecule being studied. Okay, so these are the limitations. So statement number C, which says that it can be used to calculate, it can be used to calculate the electronic properties, it is false. Okay. Now let me go to the next problem. Problem number 4 says which of the following operations or calculation cannot be done using the quantum mechanics. Your options are number A, dipole moment calculation, number B, transition state geometries and energies, number C, energy calculation for specific conformation, and number D, bond dissociation energies. Which of the following operations or calculation cannot be done using the quantum mechanics? Number A, dipole moment calculation. Number B, transition state geometries or energies. Number C, energy calculations for specific conformations. And number D, bond dissociation energy. Which of the following cannot be done using quantum mechanics? Let me know A, B, C or D. Which of the following will be the correct answer? After a minute, I will check the chat box. Okay. Which of the following operations or calculation cannot be done using quantum mechanics? Dipole moment calculation, transition state geometries or energies, energy calculation for specific conformation, or bond dissociation energies. Let me check the chat box. Let me see if there is any answer for our problem number 4. Okay. Number D, number B, number A. So I have received three different answers, number A, B, and D. Okay. So three different option as answer. Let me now check which of the following will be the correct answer. Number A, number B, and number D. I think I have received. Okay. So the correct answer will be, it is actually number C. Okay, energy calculation for specific conformation. This cannot be calculated, or it is cannot be done with the help of the quantum mechanics. Now let us understand what is molecular mechanics, what is quantum mechanics, what are the differences, and what is the choice of method for each and every type of calculation. Okay, so the various operation carried out in molecular modeling involve the use of the programs or algorithms which calculate the structural property data of the molecule in question. For example, it is possible to calculate the energy of a particular arrangement of the atoms, conformation, modify the structure to create an energy minimum, and calculate the properties such as charge, dipole moment, heat of formation, the computational methods that are used to calculate structure and property data can be split into two categories: molecular mechanics and quantum mechanics. Molecular mechanics, the equations are used which follows the law of classical physics and apply them to nuclei without consideration of the electron. Okay, that we have already discussed in our last slide. In essence, the molecule is treated as a sphere or connected with a spring, which are the bonds. So the sphere are the atom and the springs are the bond. Equation derived from the classical mechanics are used to calculate the different interaction and energies or force field resulting from the bond stretching, angle bending and non-bonded interaction, and the torsional energies. Torsional energies are associated with the atom that are separated by each other with the three bonds. Okay, the torsional energies are associated with the atoms that are separated by each other by three bonds. The relative orientation of these atoms are defined by the dihedral or torsional angle. These calculations require data or parameters that are stored in the tables now within the program which describe the interactions between the different sets of atoms. The energies are calculated by molecular mechanics, have no meaning as absolute quantities but are useful while comparing different conformation of the same molecule. Molecular mechanics is fast, less inexpensive. Okay, so it is the molecular mechanics is fast and is less intensive on computer time than the molecular quantum mechanics. However, it cannot calculate the electronic properties because electrons are here not included in the calculation. That we have already discussed a little bit in our previous class. Now let us discuss what is the quantum mechanics. So what is done in the quantum mechanics? So it is uses the quantum physics to calculate the properties of the molecule by considering the interaction between the electrons and nuclei of the molecule. Okay, so in the molecular mechanics, the electrons are not considered. Okay, now only atoms and the bonds which okay, but in case of the quantum mechanics, the electrons and the interaction between the electron and the nuclei are considered. Unlike molecular mechanics, atoms are not treated as solid sphere. In order to make the calculation feasible, various approximation has to be made. So what are the approximations in the approximations in the quantum chemistry? The nuclei is regarded as motionless. This is responsible as the motion of the electrons is much this is reasonable as the motion of the electrons is much faster. In comparison, as electrons are considered by moving around the fixed nuclei, it is possible to describe the electronic energy separated from the nuclei energy. Then it is assumed that the electrons move independently of each other, so the influence of other electrons and nuclei are taken as an advantage. Quantum nuclei mechanical can be subdivided into two broad categories. One is the ab initio and another is the semi-empirical. Okay, the former is the more rigorous and does not require any stored parameters or data. However, it is expensive on computer time and restricted to small molecules. Semi-empirical method compute for the valence electron only. They are quicker, less accurate, and can be carried out on the larger molecule. There are various forms of the semi-empirical software. For example, MINDO3, MNDO, MNDO DAM1, and PM3. These methods are quicker because they are use approximation to make use of the stored parameter. Now the choice of method. It is important that when to use the molecular mechanics and when to use the quantum mechanics. The method of calculation is chosen depends on what calculation needs to be done as well as the size of the molecule. As far as the size of the molecule is concerned, ab initio method are limited to the molecule containing 10 of the atoms. Semi-empirical calculation are contained 100 of atom, and molecular mechanics and the thousands of atoms. This is important. Okay, ab initio calculation, which is a part of the quantum calculation, are limited with the molecules containing the 10 of the atom. Semi-empirical are considered 100 of atom, and molecular mechanics containing the thousands of atoms. So this is the number of atom, so which determine which method to use. Okay, so then molecular mechanics is useful in the following parameters, which is the energy minimization, identification or interface stable conformation, energy calculation of the specific conformation. That was in our question, right? That's energy calculation for the specific conformation. So this is done by molecular mechanics, not quantum mechanics. Generating different conformations, studying molecular motion. So all these can be done using the molecular mechanics. And quantum mechanics methods are suitable for calculating the following: the molecular orbital energies and coefficient, heat of the formation of specific conformation, partial atomic charges calculated from molecular orbital coefficients, electrostatic potential, dipole moments, transition state geometries and energies, bond dissociation energies. So all these can be calculated with the help of the quantum mechanics. Okay, so these are the molecular details of the molecular and quantum mechanics and their specifications, where to use which method and what is the I mean the plus point, what is the minus point of each and every method, that is discussed here. Okay, I will suggest once I share this PPT, go through the material well, okay. And then if you have any doubt or any query, let me know in the follow upcoming class, or you can let me know before that. You can let me know via email, and I will discuss it in our upcoming class in detail. Okay, if you have any query. But first, please go through the slide when I will share it after today's class, and I think you will understand. You don't need to go to very details of the molecular or quantum mechanics, but you need to understand when you have to use this molecular mechanics and when you have to use the quantum mechanics. Okay, and what are the limitation of each and every method. Now I will go to the next problem. Problem number 5 says, which of the following statements is incorrect regarding the microwave technology? Your options are: number A, it requires shorter reaction time. Number B, coupling reactions cannot be performed using this. Number C, it leads to fewer side effect reaction. And number D, it is employed in library synthesis. Which of the following statements is incorrect regarding the microwave technology? Number A, it requires shorter reaction time. Number B, coupling reactions cannot be performed using this. Number C, it leads to fewer side effects. Number D, it is employed in library synthesis. Which of the following statements is incorrect regarding the microwave technology? Again, I will wait for a minute. Let me know A, B, C or D. Which of the following will be the correct answer for problem number 5? Which of the following statement is incorrect using the regarding the microwave technology? Which of the following statement is incorrect regarding microwave technology? Let me check the chat box. Okay, I have received one answer which is saying that number D is the correct answer. Okay, let me check if number D is the correct answer or not for our problem number 5, right? So, the statement which is incorrect regarding the microwave technology will be number B. Coupling reactions cannot be performed using this. This statement is false. Okay. So, microwave technology is a very expensive process, and microwave assisted organic synthesis is proving to be key useful tool for the accelerating synthesis and making process more efficient. Okay, there are many examples for the thermal reaction that take several hours to complete using heaters and oil baths which are carried out in minutes using the microwave condition. There is a much greater efficiency of energy transfer using the microwave technology, which accounts for the faster reaction time. Moreover, yields can sometimes be improved dramatically with less decomposition and fewer side reaction. Specially designed for microwave units are now commonly employed in the library synthesis also. For example of the reaction that have been carried out using the microwave technology include formation of amides from acid and amine without the need of the coupling reagent. Okay, metal catalyzed Suzuki coupling reaction that perform eventually with the unreactive aryl chlorides also, and metal mediated reduction and amination. The reduction that can be the reduction use 24 hours of the conventional heating and only 15 minutes in the microwave heating. Okay, which is this one. So these are the examples which can be done, the reaction that can be done using the microwave technology. First is the amide coupling. Okay, so whenever we have discussed about amide coupling, I have told you that amide couplings are not that easy to perform. You need high temperature, sometimes greater long reaction time and expensive coupling reagents for the amide bond formation. But in microwave, in less reaction time, without any coupling reaction, this amide bond can be formed easily. Also, coupling Suzuki coupling reaction, which is basic more, which is often carried out with the bromide, aryl bromides and iodides, so more efficiently, but not with the aryl chlorides. But in microwave technology, it can be done very easily within 5 minutes. Also, the reduction, this reduction which takes usually 24 hours for the conventional heating, but it takes only 15 minutes using the microwave technology. Okay, so these are the examples which can be used in the microwave technology. So whatever the, so let us now check the options. So number A, it requires shorter reaction time, which is true. Coupling reaction cannot be performed using this reaction. This is false because we have already seen that amide coupling, Suzuki coupling, all the coupling reactions can be done with the microwave technology. It leads to fewer side effects. This is also true. It is employed in the library synthesis. This is also true because this is a very, as for nowadays, it is a very important tool in the organic chemistry. It is used in the library synthesis. But one drawback of this technique is that it is very expensive. Okay. So this is about the microtechnology. Now problem number 6 says which of the following is the privileged scaffold for the medicinal chemistry? Your options are number A, benzodiazepine, number B, hydantoin, number C, tetrahydroisoquinoline, and number D, all of the above. Which of the following is a privileged scaffold for medicinal chemistry? Benzodiazepine, hydantoin, tetrahydroisoquinoline, quinoline, or all of the above? Let me know A, B, C, or D. Which of the following will be the correct answer? Which of the following is a privileged scaffold for medicinal chemistry? Benzodiazepine, hydantoin, tetrahydroisoquinoline, or all of the above? Which of the following is a privileged scaffold for medicinal chemistry? Benzodiazepine, hydantoin, tetrahydroisoquinoline, or all of the above? What will be the correct answer for our problem number 6? Let me check the chat box. Okay. So I have received two answers. Okay, I have received three answers and all are saying that number D is the correct one. Okay, let me check if number D is the correct answer or not. So the correct answer will be yes, number D, all of the above. All the scaffolds, benzodiazepine, hydantoin, tetrahydroisoquinoline are known as the privileged scaffold for the medicinal chemistry. Okay, so many scaffolds are constructed using the synthetic route employed for the solid phase synthesis, and this also determines the number or variety of substituent that can be attached to the scaffold. Okay, the ideal scaffold should be smaller in order to avoid variation of the substituent. So it should have also substituent widely dispersed around the structure rather than restricted to one part of the structure. Okay, so the structure that has the substituent all around the structure are known as the spiral-like rather than restricted to one part, which is the tadpole-like. If the conformation space around is fully explored, finally, synthesis should allow each of the substituent to be varied independently to each other. Scaffolds can be flexible, for example, peptide backbone, or rigid like a cyclic system. This may contain heteroatoms and capable of forming useful bonding interaction with the binding site, or they may not. Some scaffolds are already found in medicinal chemistry like benzodiazepine, hydantoin, tetrahydroisoquinoline, benzyl sulfonamide, and biaryls are associated with the diverse range of activities. Such scaffolds are known as the privileged scaffold. So these are the examples of the privileged scaffold. We have the dipeptide, we have the glucose, we have the steroid. Here the particular structure which is shown in blue, this is actually at the privileged scaffold, and the black one are the substituent. Okay, they may vary, but the scaffold which is shown in blue is it will remain same. Okay, this is the steroid, hydantoin, indole, lactam, pyridine, 1,4-benzodiazepine, biphenyl pyrimidine, diphenyl methane. So all these are examples of privileged scaffold. So benzodiazepine, hydantoin, beta-lactam, and pyridines are example of extremely good scaffold. Okay, so among all the privileged scaffold, these four, benzodiazepine, hydantoin, beta-lactam, and pyridine, are examples of extremely good scaffold. Why? Because they have small molecular weight, and there are various synthetic routes available which produce the substitution pattern required for the fully explore the conformation space about them. For example, it is possible to synthesize benzodiazepines such that there are variable substituent around the whole structure. Peptide scaffolds are flexible scaffolds which have the capacity to form the hydrogen bonds with the target binding sites, and they are easy to synthesize, and a large variety of different substituents are possible by using the amino acid building block. Further substitution are possible on the terminal amino and carboxylic acid function. The substituents are widely distributed along the peptide chain, allowing a good exploration of the conformation space. If we consider Lipinski's rule of 5, the peptide scaffold should ideally be restricted to di or tripeptide in order to keep the molecular weight below 500. It is interesting to note that the orally active antihypertensive agents captopril and enalapril are dipeptide, like whatever large peptides such as the enkephalin are not orally active. Oral activity has been also a problem with HIV protease inhibitor that have molecular weight over 500. Some of the scaffold shown here have a variety of disadvantages. Although glucose has a small molecular weight and possibly 5 variable position substituent, it is contain multiple hydroxy group. Attaching different substituent with a similar group usually requires complex reprotection strategies. Okay, so the problem with the glucose scaffold is that all the groups, as you can see, have this hydroxyl moiety. So so if you want to specifically functionalize this position, you have to protect all the other positions. So it requires a very complicated reprotection strategy to be followed. Nevertheless, the potential of sugar-based drug is great, and a lot of progress had been made developing solid phase synthesis based on the sugar scaffold. Steroids might appear attractive scaffold. However, the molecular weight of the steroids chemical limits the size and number of substituents which can be added if we wish to keep overall molecular weight below 500. The indole scaffold shown suffers a disadvantage in having variable substituent located in the same region of molecule, preventing a full exploration or conformation space. Because of this, this is a tadpole-like scaffold. So basically, the indole it has a tadpole-like structure because the only this position can be explored, not this the other positions can be, this region can be fully explored. Okay, so which looks like a tadpole-like structure. So these are about the privileged structures, and it will be good if you can remember a few of the structures because every now and then you have to, I mean, deal with these structures when you are reading the medicinal chemistry. And since now we are moving forward toward various therapeutic agents, you will often see this kind of structures very now and then. Okay. Now let me go to the next problem. So problem number 7 says, which of the following is not true for bacteriostatic agents? Your options are: number A, the agents stop the growth of bacteria. Number B, the bacterial population remains the same. Number C, the agents destroy the bacteria. Number D, the agents inhibit processes like replication, translation, transcription in the bacteria. Which of the following statements is not true for bacteriostatic agents? Your options are: number A, the agents stop the growth of the bacteria. Number B, the bacterial population remains same. Number C, the agents destroy the bacteria. And number D, the agents inhibit the processes like replication, translation, transcription. Which of the following statement is not true? Let me know, A, B, C or D. Again, I will check the chat box after a minute or two. Okay, let me know, A, B, C or D, which of the following statement is not true regarding the bacteriostatic agents. Which of the following statements is not true for bacteriostatic agents? Number A, the agent stop the growth of the bacteria. Number B, the bacterial population remains the same. The agent. Number C, the agents destroy the bacteria. Number D, the agents inhibit the processes like replication, translation, and transcription. Let me check the chat box now. Okay. Number C, number C. So I have received two answers and both are saying that number C is the correct answer. Okay, let me check. So the correct answer will be yes, this is number C, which says that the agents destroy the bacteria. This statement is not true for the bacteriostatic agent. Now let us understand what is a bacteriostatic and what is so two type of so the antimicrobial agents can be majorly divided into two parts. One is the bactericidal and another is the bacteriostatic. What is the bacteriostatic and what is the bactericidal? So bacteriostatic is the antimicrobial agent that reversibly inhibit the growth of the bacteria. Okay, like tetracycline and chloramphenicol are other example. And bactericidal are those which has a lethal action on the bacteria. Okay, and this is the irreversible. So bacteriostatic is that the compound that reversibly inhibit the growth of the bacteria. If you give a bacteriostatic agent, it will lower the growth of the bacteria and eventually it will inhibit all the processes like replication, replication or your translation, transcription. This in the bacteria. Okay, they will stop the growth of the bacteria. But in case of the bactericidal, the agents will kill the bacteria. Okay, so this is the difference between the bacteriostatic and bactericidal. So the bactericidal refers to the antibiotic that kill the bacteria, whereas the bacteriostatic refers to the antibiotics that prevent the growth of the bacteria. Okay, in case of the bactericidal, the action is irreversible, but in the bacteriostatic, the agents, the action is reversible. If you, if you withdraw the particular antimicrobial agent, the bacteriostatic ones, the growth of the bacteria will be restored. Now, inhibit the particular bactericidal agent, they inhibit the cell wall formation of the bacteria, whereas the bacteriostatic inhibit the DNA replication and the protein synthesis of the bacteria. Bactericidal do not work with the immune system of the host, whereas the bacteriostatic work with the immune system of the host to prevent the growth and reproduction of the bacteria. MBC, which is refers to the concentration of the drug required to kill the 99.99% of the bacterial population, and MIC is the minimum drug concentration which is inhibits the bacterial growth. So in case of the bactericidal agent, MBC is the parameter which is actually checked, and in case of bacteriostatic, MIC is the parameter that is checked to identify the efficacy of a bactericidal agent. Example of the bactericidal agents are the beta-lactam antibiotics, cephalosporin and vancomycin, whereas the example of the bacteriostatic include the tetracycline, streptomycin, chloramphenicol and sulfonamide. Okay, so this is the difference between the bacteriostatic and bactericidal agents. So the bacteriostatic agents, they stop the growth of the bacteria, but the bacterial population remains the same, and the agents destroy the remains the same, and they inhibit the processes like replication, translation and transcription, but they do not destroy the bacteria. So this statement is false. Okay. Now let me go to the next problem. Problem number 8 says which of the following statement is false regarding the gram-positive bacteria? Your options are: number A, very much susceptible towards antibiotic; number B, they have low lipid count, lipid content; number C, peptidoglycan layer is thin and majorly single layer; number D, outer membrane is not present. Which of the following statement is false regarding gram-positive bacteria? Number A, very much susceptible to antibiotic; number B, they have low lipid content; number C, peptidoglycan layer is thin and majorly single layered; and number D, outer membrane is not present. Which of the following statement is false regarding the gram-positive bacteria? Let me know A, B, C or D. Which of the following statement is false regarding gram-positive bacteria? After a minute, I will check the chat box. Okay. So meanwhile, read all the options carefully and let me know A, B, C or D. Which of the following is the correct answer for problem number 8? The statements are: number A, which where they are very much susceptible to antibiotics; number B, they have low lipid content; number C, peptidoglycan layer is thin and majorly single layer; and number D, outer membrane is not present. Which of the following is the statement is incorrect regarding the gram positive bacteria? Let me check now the chat box. Let's see if I have received answers. Okay, number A and number C. So I have received two answers. One is number C and another is number A. Let me check which of the following will be the correct answer, number C or number A. So the statement which is false regarding gram-positive bacteria is number C, which says that the peptidoglycan layer is thin and majorly single layered. Okay, this statement is false. So the primary difference between the gram-positive and gram-negative bacteria lies in their cell wall structure, which determines how they react to a laboratory procedure called gram staining. Okay, gram-positive bacteria have a thick peptidoglycan layer that retains the purple dye, while gram-negative bacteria have a thin peptidoglycan layer and an additional outer membrane, causing them to appear pink or red. Okay, so this is important. Here, number C is said that peptidoglycan layer is thin and majorly single layer in gram-positive. So this statement is false because clearly you can see that the gram-positive bacteria have a thick peptidoglycan layer. Okay, and why this is known as the positive and negative? Because this technique by which this is done is the gram staining technique, where the gram-positive bacteria have the thick peptidoglycan layer and that retains the purple dye, and the gram-negative bacteria that have a thick peptidoglycan layer and an additional outer membrane, causing them to appear pink or red. Okay, so what are the key differences and biological differences, key structural biological differences of gram-positive and gram-negative? So gram-positive bacteria, peptidoglycan layer is thick, about 20 to 80 nanometer, and many layers are there. Gram negative bacteria, these are thin, 20 to 10, 2 to 10 nanomolar, often single layer. Outer membrane is absent in case of gram positive bacteria, but it is present and it contains the lipopolysaccharides or LPS. Gram stain color purple or blue in case of the gram positive, pink or red in case of the gram negative. Teichoic acid is present and provide rigidity to the gram positive bacteria. It is absent in the gram negative bacteria. Toxins produced primarily exotoxins in case of gram positive and primarily endotoxin in the case of gram negative. Antibiotic resistant, generally more susceptible in case of the gram positive bacteria, and gram negative generally more resistant due to the outer membrane. Clinical significances: treatment, the outer membrane of gram-negative bacteria acts as a barrier and making them harder for the many common antibiotics like penicillin G to penetrate. Pathogenicity, gram-negative bacteria are often more challenging to treat and are more likely to develop multi-drug resistance. Example, gram-positive bacteria is a Streptococcus aureus, the skin infection, Streptococcus pneumoniae for the pneumonia, and the Clostridium species. Gram-negative bacteria like E. coli for UTI, Salmonella and Vibrio cholerae for the cholera. cholera. So these are the examples of the gram-positive and gram-negative bacteria. So remember, so the important thing is that gram-negative bacteria, gram-positive bacteria are very susceptible to antibiotics. They have low lipid content and outer membrane is not present, but the peptidoglycan layer is thick. Okay, but in case of the gram-negative bacteria, the peptidomembrane layer is thin, but they have an outer membrane, and this outer membrane is the reason why this antibiotics are find it difficult to enter the gram-negative bacteria. Okay, and the gram-negative bacteria, they actually are more prone to develop the multi-drug resistant. Okay. So now I will go to the next problem. Problem number 9 says, which of the following antibacterial agents work by affecting bacterial plasma membrane permeability? Your options are number A, polymyxin, number B, penicillin, number C, glycoside, and number D, nalidixic acid. Which of the following antibacterial agents work by affecting the bacterial plasma membrane permeability? Polymyxin, penicillin, aminoglycoside, or nalidixic acid? Which of the following antibacterial agents work by affecting bacterial plasma membrane permeability? Polymyxin, penicillin, aminoglycoside, or nalidixic acid? Let me check the chat box after 2 minutes. Okay. Let me know A, B, C, or D. Which of the following will be the correct answer for problem number 9? Which of the following antibacterial agents work by affecting bacterial plasma membrane permeability? Okay, let me check the chat box. Let's see if there is any answer. Okay, I have received an answer which is saying number A is the correct answer. Okay, let me check if number A is the correct answer or not. So the correct answer will be yes, it is number A. Polymyxin are the antibacterial agent that work by affecting the plasma membrane permeability. So So there are a couple of mechanisms by which an antibacterial agent can work. Okay, so this is very important, and this figure is very important. Why? Because this figure, it is very, I mean, it will help you to remember what is the bacterial cell and the drug targets of a particular antibacterial cell. Okay, so now first let me discuss the particular thing, and then I will discuss this graph or this figure. So there are 5 main mechanisms by which an antibacterial agent can act. Okay, so there are 5 main mechanisms by which it can work. First is the inhibition of the cell metabolism. Okay, then second is the inhibition of bacterial cell wall synthesis. Third is the interactions with the plasma membrane. Fourth is the disruption of the protein synthesis, and the fifth is the inhibition of the nucleic acid transcription and replication. So these are the 5 techniques by which antibacterial agent can work, or the 5 main mechanisms. The first is the inhibition of the cell metabolism. So antibacterial agents which inhibit the cell metabolism are called the antimetabolites. These compounds inhibit the metabolism of the microorganism but not the metabolism of the host. They can do this by inhibiting an enzyme catalyzed reaction which is present in the bacterial cell but not in the animal cell. The best known example of the antibacterial agent acting in this way are the sulfonamides. It is also possible for the antibacterial agents to show selectivity against the enzymes which are present in both bacterial and the mammalian cell, as long as there are the significant differences in the structures between the two. Inhibition of the bacterial cell wall synthesis leads to the bacterial cell lysis and death. Agents operating in this way include the penicillin, cephalosporin, and glycopeptides such as the vancomycin. As animal cells do not have a cell wall, they are unaffected by such agents. Interactions with the plasma membrane. Some antibacterial agents interact with the plasma membrane of the bacterial cells to affect the membrane permeability. This has a fatal results to the cell. Polymyxin and thioredoxin work in this way. Disruption of the protein synthesis means the essential proteins and enzymes required for the cell survival can no longer be made. Agents which disrupt the protein synthesis include the rifampicin, aminoglycoside, tetracycline, and chloramphenicol. And inhibition of the nucleic acid transcription and replication prevents cell division and or synthesis of the essential protein. Agents acting in this way include the nalidixic acid and chlorpyrifene. Okay. So this figure is very important and it is very easy to depict all the anti-cellular metabolism and the agents by in this way. The first is one by one let us see. So we have this is the particular outer membrane of the which is present in the gram negative bacteria only. This is the cell wall. Okay, this is the perisplasmic phase and this is the plasma membrane. Okay, so first is the outer membrane. So then we have the cell wall. So cell wall, the disruption can be done by penicillin, cephalosporin, and cyclosporin. Then we have this membrane, like this plasma membrane, which is affected by the polymyxin. Then we have this ribosomes, which are involved in the protein synthesis, which is hampered by chloramphenicol, streptomycin, and tetracycline. Then we have this nucleic acid like material like DNA and RNA that is affected by quinolone, aminoacridine, and rifampicin. And enzymes for the cellular metabolism can be hampered by the sulfonamides. Okay, so these are the techniques by which the antibacterial agent can hamper life cycle of a bacteria. Okay, inhibition of the cell metabolism, inhibition of bacterial cell synthesis, interaction with plasma membrane, disruption of protein synthesis, and the inhibition of the nucleic acid transcription and replication. So polymyxin, what the polymyxins do? So basically the polymyxin they affect the plasma membrane of the bacterial cell to affect the membrane permeability. Okay. Now let me go to the next problem. Okay, this is the structure of polymyxin. You do not need to remember the structure. I have just given it for your understanding. Okay. So problem number 10 says augmentin is a life-saving drug and it is a mixture of number A oxacillin and ampicillin, number B amoxicillin and clavinic acid, number C oxacillin and clavinic acid, and number D amoxicillin and pyruvic acid. Augmentin is a life-saving drug and it is a mixture of number A oxacillin and ampicillin, number B amoxicillin and clavulanic acid, number D oxacillin and clavulanic acid, and number D amoxicillin and pyruvic acid. Which of the following will be the correct answer? Problem number 10. Augmentin is a life-saving mixture of which of the following drugs? Let me wait for a minute, then I will check the chat box. Okay. Augmentin is a mixture of which of the following two drugs? Oxacillin, ampicillin, amoxicillin, clavulanic acid, oxacillin, clavulanic acid, or amoxicillin, pyruvic acid. Let's see if there is any answer in the chat box. Number C, number B, number B. So I have received two number B and one number C as the answer. Okay, let me check which of the following is the correct answer. So the correct answer is number B, amoxicillin and clavulanic acid. This is the correct mixture of which is known as the augmentin. So this is the structure of the amoxicillin and this is the structure of the clavulanic acid. Okay. So basically, the clavulanic acid is the about the amoxicillin, which is the we will be discussing in our upcoming slides. Okay, let us discuss about the clavulanic acid first. Okay, so the clavulanic acid was isolated from the S. clavularius by Beckham's in 1976. It has a weak and unimportant antibiotic activity, but it is a powerful and irreversible inhibitor of the most of the beta lactamases, which means it is used as a sentry drug. What is a sentry drug and the regarding the about what is how it works we have discussed in our previous class. You can go through the PPT of the week 9 to discuss to check the sentry drugs, okay, that we have already discussed in our last class. In combination with the traditional penicillin such as amoxicillin, this allows the dose levels of amoxicillin to be decreased and also increases the spectrum of the activity. However, it should be noted that there are various types of beta lactamases. Although clavulanic acid is effective against the most of this, it is not effective against all. Clavulanic acid is also administered intravenously with the ticarcillin as timentin. The structure of the clavulanic acid was the first example of naturally occurring compound where the beta-lactam ring is not fused with the sulfur-containing ring. So most of the cases, in case of the beta-lactam antibiotics, what happens that the beta-lactam ring is fused with this sulfur ring is fused with the sulfur-containing link, like it is also present in the our amoxicillin. But in case of this particular clavulanic acid, the beta lactam ring is not fused with a sulfur containing ring, but it is a fused with a oxazolidine ring, okay, or which is a oxygen containing. The structure is also unusual and it does not have any acyl amino side chain. There is no acyl amino side chain in this position present, which is in case of normal normal beta-lactam antibiotics. Okay, the many analogs have now been made and essential requirement for the beta-active inhibition are. So this is very important. What are the essential requirement of the for the beta-lactam is inhibition? Okay, there is the strain beta-lactam ring, then enol ether, the Z configuration of the double bond. Here the Z configuration of the enol ether, if activity is reduced or not eliminated if the double bond is E. No substitution at the C6 in position. R stereochemistry of 2 and R stereochemistry of 2 and 5. Okay, carboxylic acid group. This is also thought that the 9-hydroxyl group is involved in the hydrogen bonding with the active site of the beta lactamase. Clavulanic acid is a mechanism-based irreversible inhibitor and can be classified as a suicide substrate. So this is very important. If you remember, we have already discussed about the this mechanism based reversible inhibition and the suicide substitute. And when we were discussing about these things, I think I have discussed about clavulanic acid one more time. So this is the complete this is the complete SAR of clavulanic acid. What are the requirements for the antibacterial activity, and this is known as a sentry drug with amoxicillin and the combination known as the augmentin. Okay. Now I will go to the next problem. Problem number 11 says mechanism of action of penicillin involves inhibition of a particular enzyme in the bacterial cell wall. Identify this particular enzyme. Your options are number A carboxypeptidase, number B transpeptidase, number C glycolase, number D phosphatase. Mechanism of action of penicillin involves inhibition of a particular enzyme in the bacterial cell wall. Which is what is the this particular enzyme? Number A carboxypeptidase, number B transpeptidase, number C glycolase glycosylase, and number D phosphatase. Let me know A, B, C or D which of the following will be the correct answer. Problem number 11. I will wait for a minute, then I will check the chat box. Okay. Mechanism of action of penicillin involve the inhibition of particular enzyme in the bacterial cell wall. Identify which is the enzyme that is involved in this mechanism of penicillin. Let me first check the chat box. Okay, number B is the only answer that I have received for this problem. Okay, let me check if number B is the correct answer or not. So the correct answer is yes, number B, transpeptidase is the enzyme that is involved in the mechanism of action of penicillin. So, the structure of the cell wall. In order to understand penicillin's mechanism of action, we have to first look at the structure of the cell wall, that is the bacterial cell wall and mechanism by which it is formed. Bacteria has a cell walls in order to survive the large range of the environmental conditions such as varying pH, temperature and osmotic pressure. Without a cell wall, water would continuously enter the cell as a result of osmotic pressure causing the cell to swell and burst. The cell wall is very porous and does not block the entry of water, but it does prevent the cell swelling. Animal cells do not have a cell wall, making it perfect target for antibacterial agents such as penicillin. The wall of the peptidoglycan structure, and in order it is made of the peptide and sugar unit. The structure of the cell wall consists of the parallel series of the sugar backbone containing two types of sugar, one N-acetyl muramic acid and N-acetyl glucosamine. Peptide chains are bound with the NAM sugars and it is an interesting to note that the presence of the D amino acid in this structure. In human biochemistry, there are only L amino acid, whereas the bacteria have the racemase enzyme that converts D to L to D amino acid. This is very important. Okay, in the structure of the bacterial cell wall, we can see the presence of the D amino acid. In the humans, we have only the L amino acid present, but the bacteria have the racemase enzyme which can convert the L amino acid to the D amino acid. The final stage of the cells, in the final stage of the cell wall synthesis, peptide chains are linked together by the displacement of L-alanine from one chain glycis to another. About 30 enzymes are involved in the overall biosynthesis of the cell wall, but the final cross-linking reaction which is inhibited by the penicillin. This lead to the cell framework to no longer be interlinked. Okay, as a result, the wall becomes fragile and can no longer prevent the cell from swelling and bursting. The enzyme which is responsible for the final cross-linking is known as the transpeptidase enzyme. There are several types of the enzyme that vary the character one bacterial cell to another, and they are inhibited by various degrees of the penicillin. Okay, so this is the importance of the structure of the cell wall and importance of the transpeptidase enzyme, which is responsible for the final step cross-linking, and penicillin basically inhibits this final step cross-linking. So what is the mechanism of the transpeptidase cross-linking and inhibition of penicillin? So basically in the transpeptidase enzyme, we have in the active site, we have a serine and one lysine is present here. Okay, so what happened in case of the penicillin, it comes and this serine has a nucleophilic OH which attacks to the beta-lactam ring, and it is bind with the active site of the transpeptidase enzyme. Once it is bind, the peptide chain cannot bind to the active site and it is blocked, and that's how the inhibition happens. Okay, so what is the structure of penicillin? This is the structure of penicillin, which is the that I have mentioned in the last slide that I will discuss later. So we have a 4-member beta-lactam ring. Okay, and also we have a 5-member sulfur containing ring, which is the thiazolidine ring. Okay, this is the structure of the 6-APA or 6-amino penicillinic acid up to this. Okay, and there we have a acyl side chain in this position present. Now when R is the this CH2 phenyl, it is known as the benzyl penicillin on penicillin G, and when it is the OCH2PH, this is known as the phenoxymethyl penicillin or penicillin V. Okay, so this is the structure of the penicillin, and this is the how the mechanism the particular blocking happens. Okay, now let me go to the next problem. Problem number 12 says, which of the following is an incorrect statement regarding the SAR of penicillin? Your options are: number A, beta-lactam ring is essential; number B, a bicyclic system is essential; number C, methyl groups in C2 position are essential; number D, free carboxylic group is essential. Which of the following statement regarding SAR of penicillin is incorrect? Number A, beta-lactam ring is essential. Number B, a bicyclic system is essential. Number C, methyl groups in C2 position is essential. And number D, free carboxylic acid group is essential. Let me know A, B, C or D, which of the following will be the correct answer for problem number 12. Which of the following is incorrect statement regarding the SAR of penicillin? Number A, beta-lactam ring is essential. Number B, a bicyclic system is essential. Number C, methyl groups in situ position is essential. Number D, free carboxylic acid group is essential. Okay, let me check the chat box now. Let's see if there is any answer for our problem number 12. Okay, I have received one answer, which is number C. Okay, let me check if number C is the correct answer or not. Okay, 1 second. Yeah. So, the correct answer will be yes, number C. Methyl groups in C2 position are essential. This statement is false. So, SAR of penicillin is a very important, very important topic. Okay, and you have to understand and you have to remember. Okay, the what is the important structural activity relationships in the penicillin of the penicillin. So, this is the structure, the broad structure of penicillin. The numbering starts from the sulfur 1, 2, 3. 4 is the nitrogen, 5 and 6 is the this position. Okay, and 7th is the this position. This is a beta lactam ring I have already discussed, and this is a thiazolidine ring. So this lactam ring is essential. The bicyclic system is also essential. Okay, the cis stereochemistry of these two hydrogens of 5 and 6 position are essential. This aramide side is also essential, and the free carboxylate is also essential. But the methyl groups are not essential for the structure, but all these things are essential. And so the large number of penicillin analogues have been synthesized and studied. The results of these studies lead to the following SR conclusion. The strain beta-lactam ring is essential. The free carboxylic acid is essential. This is usually ionized. The penicillins have administered as the sodium or potassium salts. The carboxylate ion binds to the charged nitrogen of the lysine residue in the binding site. The bicyclic system is important. This confers further strain on the beta-lactam ring. The greater is the strain, the greater is the activity, and the greater is the instability of the molecule to other factors. The acyl amino side chain is essential. Sulfur is usual but not essential. The stereochemistry of the bicyclic ring is respect to the acyl amino side chain is also important. These results of the analysis led to the inevitable conclusion that the very little variation is tolerated by the penicillin nucleus, and that any variations are restricted to the acyl amino side chain. Okay, so you can change this side chain, this R position. It can be changed. This is tolerable, the changes in this R group, but not the other things. Okay, so whenever if you are asked to do so highlight the SAR, what is the important, what is not, so you have to remember the points. Okay, the points as well as try to draw the structure figure. Okay, the figure is very I mean it is not that much hard. If you practice it 1 or 2 times, the structure is not that much I mean difficult to understand, and these things are very actually this figure is very nicely, this has described all the SAR of the depict all the SAR of the penicillin. Okay, like the importance of the beta lactam ring, the stereochemistry of this position, the bicyclic system, the amide side chain of this sixth position. Okay, free carboxylate group, these are also important. Only change that can be made to the this is the R group. Okay, so this is the SAR of the penicillin. Now, problem number 13 says erythromycin belongs to dash class of antibiotics. What will be the correct answer? Number A, macrolide, number B, tetracycline, number C, oxalidione, and number D, quinolones. Erythromycin belongs to which class of antibiotic? Macrolide, tetracycline, oxalidione, or quinolone? Again, after a minute, I will check the chart box. Let me know A, B, C, or D, which of the following will be the correct answer. Erythromycin belongs to dash class of antibiotic. Macrolide, tetracycline, oxalidione, or quinolone? Let me know. Let me check the chat box. Let's see if we have any answer for problem number 13. Okay, I have received an answer which says that number A is the correct one. Let me check if number A is the correct answer or not. The correct answer will be yes, number A. Erythromycin belongs to macrolide class of antibiotics. So macrolides are the bacteriostatic agents. Okay, so this is important to remember that the macrolides are the bacteriostatic agents. They best known example of the class of the compounds are erythromycin. So the structure of the macrolides are very difficult. Okay, I mean they are very complicated structure. You do not need to memorize the structure. Okay, neither you need to practice it. Just remember how it looks. Okay, because if the question is given with the structure, so that you can identify that it is a macrolide and you can answer that question correctly. Okay, so as you can see, this is the structure of a macrolide. The blue portions are important, like this lactone ring in this position, the sugar, this is the clarinose, and this is the disaminose sugars, and in this position is important. So this is the erythromycin when the x equal to OH. Okay, when the x equal to OH, it is the erythromycin, and when the x equal to OME, it is the calithромycin, right? This is the structure of azithromycin. The azithromycin is a very well known antibiotic that we take, and then we have also have the telithromycin, which has a very unusual structure. Okay, so the best known example of the class of macrolide is the erythromycin, a metabolite isolated in 1952 from the soil microorganism Streptomyces erythrinaceus, found in the Philippines, and one of the safest antibiotics for the clinical use. The structure consists 14-membered macrolide lactone ring with a sugar and the amino sugar attached. Okay, this is important. This you can remember, like that it is a 14-member macrocyclic lactone with a sugar and amino sugar. The sugar residues are important for the activities. Okay, the sugar residues which are present in the erythromycin or the macrolides, it is important for its biological activity. Erythromycin acts by binding to the 50S subunit of the bacterial ribosome to inhibit the translocation, but other mechanism of action are appear to be likely because erythromycin and chloramphenicol bind to the same region of the ribosome. They should not be administered together as they will compete with each other and be less effective. Erythromycin is unstable to stomach acid but can be taken orally in a tablet form. The formulation of the tablet involves a coating that is designed to protect the tablet during the passage through the stomach, but which is soluble once it reaches to the intestine. The acid insensitivity and erythromycin is due to the presence of the ketone and two alcohol group, which are set for the acid catalyzed intramolecular formation of the ketal. One way of preventing this to protect the hydroxyl group, for example, clarithromycin is a methoxy analog of erythromycin, which is more stable and to the gastric juices and has improved the oral absorption. Another method of increasing the acid stability is to increase the size of the macrocycle to a 16-member ring. Azithromycin contains 15-member macrocycle where the N-methyl group has been incorporated into the macrocycle. It is one of the world's best-selling drugs. Telithromycin is a semi-synthetic derivative of erythromycin, reached to the European market in 2001. The clarinose sugar in the erythromycin has been replaced with the keto group and carbamate ring to be fused with the macrocyclic ring. Okay, so here you can see this macrocyclic ring. So the amino sugar of the erythromycin is replaced by this particular the group, and this is fused with this macrocyclic ring. Okay, the two hydroxyl group can cause intermolecular ketone formation in the erythromycin have been marked as the methoxy group, and another is the carbamate ring. Okay. So this is all about the macrolide. So the important thing that you have to remember the examples of the particular, and whenever I am discussing about a antibiotic class, so you have to remember the examples. Okay, how the structure looks overall and what is the mechanism of action. Like erythromycin, it hinders the protein synthesis by binding with the 50th subunit of the bacterial ribosome to inhibit the translocation. This is the mechanism of action. Okay. Now I will go to the next problem. Problem number 14 says how oxalidione work. Your options are number A, it inhibits the protein synthesis by binding to the 30S ribosome. Number B, it inhibits the protein synthesis by binding with the 50S ribosome. Number C, it unwinds the DNA double helix. And number D, it inhibits the DNA guidage enzyme. How oxalidione work? Number A, it inhibits the protein synthesis by binding to the 30S ribosome. Number B, it binds with the inhibits the protein synthesis by binding with the 50S ribosome. Number C, it unwinds the DNA double helix, and number D, it inhibits the DNA guidage enzyme. Which of the following will be the correct answer for problem number 14? Let me know. A, B, C or D, which of the following will be the correct answer? Let me check the chat box. Okay, number B is the given answer. Let me check if number B is the correct answer or not. So the correct answer will be yes, number B. It inhibits the protein synthesis by binding with the 50S ribosome. So oxalidione, this is the structures of the oxalidione, this is the linazoid, and this is the radizolid. Okay, so these are the example of the oxalidinones. So oxalidinones are the new class of synthetic antibacterial agent discovered in recent years. They inhibit the protein synthesis at a much earlier stage than previous agents and consequently do not suffer the same resistant problem. Before protein synthesis can start, a 70S ribosome has to be formed by the combination of a 30S ribosome with a 50S ribosome. The oxalidinone bind with the 50S ribosome and prevent this from happening. Okay, so this is important. What does this particular oxalidinone do? They bind with the 50S ribosome and prevent the combination of the 50S and 30S ribosome to form the 70S ribosome in the bacterial cell. Okay, that as a result, translation cannot even start. Other agents can inhibit the protein synthesis during the translation process itself. Linazolid was the first of this class of compound to reach the market in 2000, and by 2010 it was netting sales of the 716 million per year. Extracrystallographic studies have revealed that how the structure binds with the ribosome, and it has allowed the development of analogs which bind more strongly. Radizolid is one of the such structure which binds 10,000 times more strongly as a result of the extra binding interaction, like extension strategy what we have discussed earlier. So the oxalidinone has a broad spectrum activity and are active against bacterial stain which are which are acquired resistant to other antibacterial agent acting against the protein synthesis. Linazolid has a good bacterial activity against the most clinically important gram-positive bacteria including MRSA. It can also be taken orally. It can also be taken orally with 100% uptake from the GI tract. Unfortunately, there is a high level of side effect related to the use, and it is bacteriostatic agent and a greater risk of the bacterial resistant development. Okay. This is all about the oxalidinone. Problem number 15 says dash acts as a starting material of the 3-methylated cephalosporin. What will be the correct answer? Your options are number A sulfonamide, number B streptomycin, number C penicillin, and number D erythromycin. Which acts as a starting material of 3-methylated cephalosporin? Sulfonamide, streptomycin, penicillin, or erythromycin? You have to fill in the blanks. Dash acts as the starting material of 3-methylated penicillin. What will be the correct answer? Number A sulfonamide, number B streptomycin, number C penicillin, and number D erythromycin. Again, let me wait for a minute, then I will check the chat box. Okay, let me know A, B, C, or D, which of the following will be the correct answer of problem number 15. Dash acts as a starting point or starting material of the 3-methylated cephalosporin. Let me check the chat box. Let's see if there is any correct answer or any answer in the chat box for our problem number 15. Okay, so I have received one answer. Okay, I have received three answers actually. One is number A, then number C, and number C. So two number C and one number A I have received as an answer. Let me check. So the correct answer is number C, penicillin, which acts as a starting material for the 3-methylated cephalosporin. Okay. So cephalexin has a methyl substituent at the position 3 which appears to help with the oral absorption. Okay, a methyl group would normally be bad for the activity because it is not a very good living group. However, the presence of the hydrophilic amino group in the alpha carbon of the 7-acyl amino side chain of cephalexin helps to restore the activity of the and the cephalexin is one of the few cephalosporin which is orally active. The mechanism of absorption through the gut wall is poorly understood. It is not clear why the 3-methyl group is advantageous for the absorption. So the synthesis of the 3-methylated cephalosporin involves the use of penicillin as the starting material. Okay, so this is a penicillin which is acts as a starting material. Okay, the synthesis which was first demonstrated by the Eli Lilly pharmaceutical involving oxidation of the sulfur followed by the acid catalyzed ring expansion where the 5-member ring thiazolidine ring in the converted to 6-member dihydrothiazine ring of the cephalosporin. So at first is the oxidation of this sulfur in this position which happens, then followed by the ring expansion. Okay, then this the ring opening happens here and the formation of the double bond. After that there is a expansion of the ring and the 6-member ring is formed which is the in the cephalosporin. So this is the 3-methylated cephalosporin and the starting of this in case this 3-methyl group actually is one of the methyl group is one of this methyl of the 2 methyls of the penicillin. One methyl group the carbon is involved in the ring expansion and another it forms this 3-methylated cephalosporin. And the 3-methylated cephalosporin, the important example is the cephalexin among the few cephalosporin which is orally active. Okay, so remember the as a synthesis of the 3-methylated cephalosporin, penicillin acts as a starting material. Now problem number 16, which of the following antibiotics work by targeting DNA guidase? Your options are number A sulfonamide, number B tetracycline, number C quinolone and number D glycopeptides. Which of the following antibiotic acts as a targeting DNA guidase? Sulfonamide, tetracycline, quinolone and glycopeptides. Which of the following antibiotics work by targeting DNA guidase? Sulfonamide, tetracycline, quinolone and glycopeptide. Let me know A, B, C or D. Which of the following will be the correct answer? Which of the following antibiotics work by targeting DNA guidase? Sulfonamide, tetracycline, quinolone, or DNA or glycopeptides? Let me check the chat box because okay, I think a minute has already passed. Okay, so I have received the answer number C, number B, number C, and number A. So three different answers again, number B, C, and number A. Let me check which of the following will be the correct answer for problem number 16. So the correct answer will be, it is number C, quinolones. Okay. So quinolones are the class of antibiotic that work by targeting the DNA guidage. So this figure I have already mentioned earlier also, and this is a very good figure actually to showcase all the antibacterial agents in a one diagram. So the three major mechanism of actions which can be exploit in case of the antibacterial drug discovery. First is the cell wall synthesis. Okay, in the cell wall synthesis, the beta-lactam antibiotics are there, penicillin, cephalosporin, carbapenem, monobactam, vancomycin and bactricin are also there, and polymyxins which attacks on the cell membrane. Then we have the protein synthesis. Already we have discussed about the macrolides which targets the 50S ribosome, clindamycin, linezolid, chloramphenicol, chloramphenicol and streptogramcin. 38 subunits are targeted by tetracycline and aminoglycosides. Then we have this nucleic acid synthesis, the blocking of nucleic acid synthesis. So this can be done in various manner. So DNA guidase are one of these. DNA guidase is a very important enzyme that can be inhibited by the quinolones. DNA polymerase can be inhibited by rifampicin, and folate synthesis can be inhibited by the sulfonamides and trimethoprim. Okay, so DNA guidase is an essential bacterial enzyme that catalyzes the ATP-dependent negative supercoiling of the double-stranded closed circular DNA. So the DNA guidase is basically the enzyme which is similar to our in the normal healthy human cell. The enzyme class of group of classes are known as the topoisomerase, and I think I have discussed it earlier also, that helps to relieve the supercoiling of the DNA during the replication. That is the DNA topoisomerase in our humans, in our human body, but in bacterial system their enzymes are known as the DNA guidase. So these are the very essential bacterial enzyme that catalyzes the ATP-dependent negative supercoiling of the double-stranded closed circular DNA. Guidase belongs to a class of enzyme known as the topoisomerase that is involved in control of the topological translation of DNA. Topoisomerase are able to change the DNA topology to facilitate the functions such as the DNA replication and transcription. Okay, so this is the structure of the quinolones. So the quinolone bind to the DNA guidase complex using the DNA replication. So this is the structure. Quinolones are the bactericidal. So this is important. Okay, to remember that this quinolone, they are bactericidal. Okay, they inhibit the bacterial DNA synthesis and several ways, causing the rapid cell death. Okay, so the quinolones, they are bactericidal. They causes rapid cell death because they inhibit the bacterial DNA synthesis in several ways. Okay, and this is the structure. So as you can see, there are several parts by which we can divide the molecule. This particular part is the stacking domain. This particular part interacts with the DNA. So this is the DNA binding domain. This is the binding to the enzyme, and this also the binding to the enzyme. So the sides binds to the enzyme, the this portion binds to the stacking domain, and this is the binding to the DNA. Okay, so this is all about the quinolone. So quinolone bind with the DNA guidage complex, blocking the further DNA replication. Now I will go to the next problem. Problem number 17 says rifampicin binds to dash. Fill in the blanks. Your options are number A, RNA independent DNA polymerase, number B, RNA dependent DNA polymerase, number C, DNA independent RNA polymerase, and number D, DNA dependent RNA polymerase. Rifampicin binds to which of the following enzyme? Number A, RNA independent DNA polymerase, number B, RNA dependent DNA polymerase, number C, DNA dependent RNA polymerase, and number D, DNA dependent RNA polymerase. Let me know A, B, C or D, which of the following will be the correct answer for problem number 17. Rifampicin binds to which of the following enzyme? Number A, RNA dependent DNA polymerase, number B, RNA dependent DNA polymerase, number C, DNA independent RNA polymerase, and number D, DNA dependent RNA polymerase. Let me know A, B, C or D, which of the following will be the correct answer. Let me check the chat box. Okay, number D, number C, number B. So I have received three different answers again. Number B, number C, and number D. Let me find out which of the following will be the correct answer. So the correct answer is number D. Rifampicin binds to DNA dependent RNA polymerase. Okay, in the last slide already I have shown you that part, that it is a RNA polymerase inhibitor. Okay, so this is the structure of basically this is the structure of rifampicin B. When the R equal to hydrogen, it is the structure of the rifampicin B, and when the R equal to this group, it is the rifampicin. Okay, and this is the rifaximin. The rifamycin is a semi-synthetic rifamycin made from the rifamycin B, an antibiotic which is isolated from Streptomyces mediterranean in 1957. It inhibits the gram-positive bacteria and works by binding to the non-covalently to the DNA-dependent RNA polymerase and inhibiting the start of the RNA synthesis. The DNA-dependent RNA polymerase in the eukaryotic cell are affected because of the drug binds to the peptide chain not present in the mammalian RNA polymerase. It is therefore highly selective. A flat naphthalene ring and several other hydroxyl groups are essential for the activity. So this naphthalene ring has several hydroxyl groups are important for the activity. Okay, and the molecule exists in a zwitterion, giving it a good solubility in lipids and aqueous acid. Rifampicin is another semi-synthetic analog that was approved in 2004 for the treatment of diarrhea and E. coli infection. So the important thing that you have to remember, apart from that, it is that it binds with non-covalently with the DNA-dependent RNA polymerase and inhibits the RNA in the start of the RNA synthesis. Okay, is that what is the SAR of this ring? So because it is therefore highly because so as I have already mentioned that it is highly selective. A flat naphthalene ring and several hydroxyl groups are essential for the activity, and the molecule exists as a zwitterion, giving it a good solubility in both lipids and the aqueous acid. Okay, so this is the naphthalene ring which is present in the rifampicin, which is important, and also the hydroxyl groups are very much important. And as it is also mentioned that the molecule stays in the zwitterion fashion. Okay, so these are the important SAR. Again, the remembering the structure is not important, but try to remember how it looks, that what are the essential parts of the structure that is important for the activity of rifampicin. Okay, now let me go to the next slide. Next problem number 18 says aminoglycosides work by blocking dash during the translation. Your options are number A tRNA binding, number B translocation, number C peptide chain transfer, and number D incoming amino acid. Aminoglycosides work by blocking which of the following steps during the translation? Number A tRNA binding, number B translocation, number C peptide transfer, and number D incoming amino acid. Which of the following is blocked by the aminoglycoside class of antibiotic agent? Let me know A, B, C or D. Which will be the correct answer for problem number 18? Aminoglycoside works by blocking which of the following during the translation? Aminoglycosides work by blocking which of the following? Let me check the chat box. Let's see if there is any answer. Okay. Number A, number B. So I have received two answers. One is saying number B, another is saying that number A is the correct answer. Okay, let me find out which will be the correct answer. The correct answer will be number B, translocation. Translocation is blocked by the aminoglycoside class of antibiotics. So basically, it's the stages at which the antibacterial agent inhibit the translation. So this is the complete translation step. Okay, so first is the 50S and 30S ribosomes. They come together to form the 70S ribosome. Here is the mRNA and there is the growing protein peptide chain, where the first is the tRNA, which brings the amino acids, and then amino acids they form the peptide chain. So now let us look at all the antibacterial agents that work by hindering the protein synthesis. First is the oxalidenone. They bind with the 50S subunit of the ribosome and binding with the 50S subunit so that the 70S subunit cannot be formed. Okay, so first is the oxalidenone. The second is the tetracycline, which blocks the tRNA from binding in this position. Okay, so the tRNA, the binding of tRNA is blocked by the tetracycline. Then we have the chloramphenicol, which blocks the peptide chain transfer. So chloramphenicol, what does they do? They blocks the peptide chain transfer. Then we have this aminoglycoside, which blocks the translocation. Okay, so aminoglycosides, they block the translocation And at the end we have the macrolides. They block the translocation. So macrolides and the aminoglycosides, these two antibacterial agents, they block the translocation, whereas the oxalidenone bind to the 50th subunit, tetracycline blocks the tRNA binding, and chloramphenicol blocks the peptide chain transfer. So these are the mechanism by which the the translation is blocked by various antibacterial agents. Okay. Now, problem number 19, it says which of the following is a second line drug for the treatment of tuberculosis? Your options are number A, isoniazid, number B, rifampicin, number C, beta-quilin, and number D, ethambutol. Which of the following is a second line drug for the treatment of tuberculosis? Number A, isoniazid, number B, rifampicin, number C, beta-quilin, and number D, ethambutol. After a minute, I will check the chat box. Meanwhile, let me know A, B, C, or D, which will be the correct answer for problem number 19. Which of the following is the second line drug for the treatment of tuberculosis? Which of the following is the second line drug for the treatment of tuberculosis? Isoniazid, rifampicin, beta-quilin, or ethambutol? Let me check the chat box. Okay. Okay, so I have received three answers and all are saying that number C is the correct answer. Let's find out. So yeah, number C is the correct answer. Bedaquiline is the second line drug for the treatment of tuberculosis. So anti-tubercular drugs are medication used for the treat infections caused by the Mycobacterium tuberculosis. Because of the bacteria can easily develop resistant, treatment always involve the combination of multiple drugs taken over several months. First line drugs, these are the most effective and least toxic medication used to treat standard drug susceptible tuberculosis. They are often remembered by the acronym HRZD. H is the isoniazid, a potent and bactericidal drug that inhibits the synthesis of the mycolic acid, a key component of the bacterial cell wall. It can cause peripheral neuropathy, which is typically prevented by taking vitamin B6 supplement. Rifampicin or R, a bactericidal drug that inhibits the bacterial RNA polymerase, a notable but harmless side effect that it turns the body fluid, urine, sweat, tears into the orange-red color. Pyrazinamide or Z, especially effective in acidic environments such as inside certain immune cell. It may increase the uric acid level, potentially leading to gout. And ethambutol, a bacteriostatic agent that prevents bacterial growth by interfering with the cell wall synthesis. A primary side effect of the neurocyte, which can cause blurred vision or red-green color blindness. Second line treatment for the drug resistant TB drugs. Second line treatment are caused by the first line drugs fail, and when dealing with the multi-drug resistant TB or extensively drug resistant TB, these are generally more toxic and require longer treatment period. For example, fluoroquinolone, including the levofloxacin and moxifloxacin, these are the now core drugs for the MDR TB due to their efficacy and oral administration. Injectable aminoglycosides like amikacin, streptomycin, formerly the first line. Newer agents specifically approved for the highly resistant TB. Bedaquiline inhibits the bacterial energy supply, ATP synthase, and delamanide and proteomide inhibit the cell wall synthesis via different pathways than first-line drugs. Other oral agents like nitinamide, cycloserine, and para-aminosalicylic acid or PAS. Okay, standard treatment regimen for now: drug-sensitive TB, WHO generally recommends the 6-month course, intensive phase of 2 months, taking all 4 first-line drugs quickly to kill the bulk of bacteria. Continuation phase or 4 months, taking only isoniazid and rifampicin to eliminate any remaining dormant bacteria and prevent the relapse. Okay. So this is the standard treatment regimen for the tuberculosis. Now, the last question of today's session, which of the following mechanisms is followed for the resistance of the sulfur-based antibiotic? Your options are number A, target mimicry, number B, efflux pump, number C, drug inactivation, and number D, target overproduction. Which of the following mechanisms is followed for the resistance of the sulfur-based antibiotic? Target mimicry, efflux pump, drug inactivation, or target overproduction? Let me know A, B, C, or D, which of the following will be the correct answer? Which of the following will be the correct answer for problem number 20? Which of the following mechanism is followed for the resistance of sulfur-based antibiotic, target mimicry, efflux pump, drug inactivation, or target overproduction? Which of the following mechanisms is followed for the resistance of the sulfur-based antibiotics? Let me check the chat box. Let's see if there is any answer. Okay. Number A, number D, and number D. So I have received two different options as the answer. One is number A and another is number D. So let me check which of the following will be the correct answer. So the correct answer will be it is number D. Target overproduction is the mechanism followed by for the resistance of the sulfur-based antibiotic. So in case of the sulfur-based antibiotics, okay, so sulfonamides generally they mimic the para-aminobenzoic acid or PABA, which is a normal substrate for the dehydroprotease synthesis. Okay, this means the sulfonamide will bind to the same manner to the para-aminobenzoic acid. So this is the para-aminobenzoic acid. This is the enzyme active site of the enzyme dehydrosteroid synthesis. Okay, and sulfonamide, what they do, they bind with the exactly same manner with this PABA to the active site, and you can see that there are so much structural similarity between these two. So the overproduction of the substrate or ligand for the target protein, overproduction of the substrate for the target enzyme will competitively block the ability of the drug to bind at the active site. For example, overproduction of PABA as the mechanism of resistance of the antibacterial sulfur drug. So it is basically how the competitive, so because sulfonamides, I think we have discussed it earlier also, that it is the competitive inhibitor for this particular enzyme, and like any competitive inhibitor, the method of how to do it, so you have to use excess of the normal substrate so that there is a competition, and if the substrate is extra, like if the target overproduction happens of the para-aminobenzoic acid, they will replace the this particular some antibiotics, and it will bind as it is. And in case of this sulfur based okay, the sulfonamide is one thing I would like to show you, that in case of the cellular metabolism, this particular mechanism is very important, like the conversion of para-aminobenzoic acid to DHFA and to THFA. This is very important. The first step is actually blocked by the sulfonamides, and the second step is blocked by the trimethoprim. That I forgot to mention that time. Okay. So this was the last question of today's session. So in today's class, what we have done, we have majorly discussed about the different antibacterial agents. Like for example, I have tried to give you an overview of each and every class, like be it macrolides, or it is a rifampicin, oxalidenone, or these are the penicillin class of antibiotics. Have tried to give you the overview of each and every class. Okay, so the most important in this particular class is, or the whenever we are discussing about therapeutic agent, is to remember what are the classes, what are the mechanism of action of each and every class, and what is the example. If it is possible to remember the structure, you can remember. Like for penicillin, it is absolutely important. I mean possible to remember the structure, and it is important. Otherwise, you can skip the structure, but remember how the structure looks, and some specific details of the structures which are important for the drug's activity. So I am sharing my email ID. If you have any doubt or any query regarding today's class or any of our previous class, you can let me know via this email. Okay, or you can you can drop your queries to the discussion forums also. So today it was our week 10 live session. So and we majorly discussed about the antibacterial agents along with some important of the computers in the medicinal chemistry, how they can be applied. Okay, so today it was our week 10 live session. In the next, so I will meet you on next Monday with our week 11 live session. There we will, I think, discuss about the antiviral agents and anti-cancer agents. That is also very important topics of the medicinal chemistry. Okay, so if there is no query, I will close the session for today. Any doubt, anything, please go through the PPT that I will submit after the end of the session. I will upload the PPTs. Please go through it, and if any query raises, please let me know. Okay. Thank you all for joining. Thank you all for answering throughout the class. Let's meet on next Monday. Okay. I will try to upload the material as soon as possible. Okay. Thank you. Thank you all.